So I'm going to start this quarter off with Chapter 7, which begins with fertilization. And before we even get into the sort of the mechanics of fertilization, I need to talk a little bit about some other things, the birds and the bees.
Mitochondria is the powerhouse of the cell.
This little thing, the cell, it's got a big journey to go on. It's going to have to pack a lot of food with it to get there to make contact with the egg. Now finally, here, this line in blue, those are the microtubules. Tubules. And they're going to help the tail move back and forth for movement. I often abbreviate them as just MT. So if I see that or I do that later in the lecture, that's why. So let's talk about the anatomy of the egg cell. It's quite a bit simpler, actually, because most of the stuff we can't really see, and we'll talk more about the stuff that's inside in just a few minutes. So anatomy of the egg cell. This is my egg cell. So this, this represents just, come on, Nance, the cell membrane. Cell membrane. And there's a layer that surrounds that cell, surrounds the cell membrane in many vertebrate organisms, and it's called the vitelline membrane. Vitelline membrane. And it's got a bunch of glycoproteins that are in there, some of which extend from the cell membrane out into that space there. The third layer is found. You see this sort of white fuzzy or gray fuzzy that I have on there? This is what's called the jelly coat, and it's not found in all organisms. can you think of which kinds of organisms might have a jelly coat surrounding them? If you thought of something sort of like organisms that have external fertilization, you would be correct. So, for example, in the xenopus, the man frog. What am I talking about? Come on, Nance. The male frog will clasp the female, and then he will fertilize the eggs as they're being deposited by the female. And because they're in sort of just out in the environment, let's say in a pond or a stream, the eggs are pretty fragile. And so the jelly coat acts sort of like a shock absorber as they sort of fall out into the environment, into the water. All right? And so that's a protective layer that's seen there. One of the cool things that you'll see in just a few minutes is typically the vitelline membrane is very, very tight against the cell membrane, and you really can't distinguish one from the other. But once sperm make contact and there's fertilization happening, there's fluid that gets kind of pumped into the space between the two membranes, and then it'll come out like a halo, and you'll be able to see it much more distinctly. All right, so that's basic. Oh, no, there's one more thing I guess I should say. That's kind of our nucleus, and I'm drawing it with the sort of dashed line just to show that, again, this is a haploid nucleus. Come on. And so it's often referred to as the pronucleus. It's not a nucleus yet because it hasn't reformed the entire nuclear membrane yet, and it's haploid, so half the number of chromosomes. All right, let's take a look about how these things interact with each other. I'm going to use a couple of different model organisms that have been used in developmental biology. And the first one that I'll talk about in terms of fertilization is going to be experiments that were done with sea urchins. I even had in my high school biology, now we're talking a long time ago, that we got to do these experiments with sea urchins. My instructor ordered them from God knows where, some biology supply place. And I think if you put them in some sort of a high salt environment, they will be induced to spawn the eggs and release sperm. And so we got to see this process happening. It was really, really cool. All right, so check this out. I learned how to do a little animation. So watch those little spermies kind of wiggle around and move. So the sperm are beginning their journey trying to find the egg. An egg, whoa, it's jumping around a little too much. The egg has been releasing a chemical that's species specific to attract sperm of the same species towards it. So here they come. They are sensing that concentration gradient of that compound by receptors in the sort of headpiece of the sperm. So let's take a look at what happens as the sperm make their way to the egg in a process that I'll call chemotaxis, or chemical attraction. Now think about this. We've got a whole bunch of different organisms that are aquatic organisms, let's say in a pond or a stream or something. And it's spring and it's reproducing time. And so you've got all these different types of organisms dumping their gametes into the water. And it's very, very important for species survival for the right sperm and the right egg to come in contact with each other to keep the species moving forward. And so the organisms have developed this way to attract, the egg has developed a way to secrete a chemical that will attract only sperm of that same species. And so let's take a look at what happens with that. So what I've got here, let me just grab my pen. So what I've got here, this kind of blue cloud, remember I was drawing that earlier. Basically what's happening is that the egg cell is releasing a chemical substance that's going to be a chemoattractant, something that's going to attract the sperm. And it was releasing it right around the jelly coat. And I'm trying to draw it as if it was a sort of diffusing away. And here comes our sperm. And it turns out that there are receptors on the surface head of the sperm here that are specific for this particular chemical. I'm not drawing them just for clarity's sake. There's receptors for the chemoattractant released by the egg. Released by the egg cell. And so I'm going to show you something that's called the acrosomal reaction. When we did the anatomy of the sperm, this little red vesicle that's sort of kind of squished up here, that's called the acrosomal vesicle, and it contains digestive enzymes in there. And so once it's going to follow, the sperm is going to swim in a sort of directed motion towards higher concentrations of the chemical that's being released by the egg. This was first discovered in sea urchins, and they characterized the chemical that was released, and they named it resact, R-E-S-A-C-T. And so other organisms that do external fertilization have their own sort of form of resact. So this sperm and all the others are trying to follow this up a concentration gradient. And the way that it does that is the more receptors, depending, let's say that there's a high density of receptors sort of right up in this hemisphere here, the head of the sperm. Well, if the ones right here, right around the midline, are receiving and binding the resact more than the ones on the edges, it's going to make a directed swim straight ahead. If, for example, the receptors that are on the side here are binding more of that resact, it's going to actually initiate a turn. I don't know if I can do this. Let me see. Hang on a sec. Let's try this. It's going to initiate a turn. Oh, no, wait. I don't want to turn the words. I want to turn the sperm. Where's my sperm? There's my sperm. No, I can't do it. It'll turn everything. Anyway, it'll make a turn so that the head of the sperm is facing this direction, and that will allow it to go swim that way. And in that sense, it's following this concentration gradient of that resact from a low concentration to a high concentration. There's cool videos, and I'll see if I can find one, where the experiments were done in a slide where they've got sperm without any egg there, and if they're observing them under the microscope, you can see that the sperm are basically swimming in random circles with no directed movement. If you add resact, sometimes you can do it in a small little sponge, and so it's going to be like the egg releasing it out as a concentration gradient. You can watch in real time as they start to recognize or pick up the scent, if you will, and then start a directed movement towards the source of that chemical attractant or that resact. Pretty cool. All right, so let's show you how that happens. So how does it know how does it like change that? Change the direction? Oh, I want to get rid of that line. Why is that line there? There, that's why the line is there. I drew it on this layer. There we go. Okay, so right when it starts to pick up the scent and move in that towards the direction, what's going to happen is, and this is something I didn't draw, just between the acrosomal vesicle and the nucleus, so this is our nucleus, is actin filaments. And at the point before it starts to make that directed movement, they're not polymerized. It's just a few of the subunits. But what will happen is, once it starts to pick up that resact and starts to move, this vesicle is going to fuse with the cell membrane here. So the acrosomal vesicle is going to fuse with the cell membrane of the head of the sperm. So what I'm drawing that as this. So this is the pink membrane representing that was what the acrosomal vesicle was. That's that membrane. And what it does is it dumps the contents, and that's what I'm showing in this sort of blurry pink color. It dumps the contents externally. So all the stuff that was in there is now on the outside. And the way it starts to make this directed movement is wherever the receptors that were receiving the highest concentration of binding of that resact, these actin filaments will polymerize, so many subunits joining together to make a polymer, and they push this the head of the sperm outward, so it makes this finger-like projection. Turns out that these contents of the acrosomal vesicle are actually digestive enzymes, which are going to allow the sperm to tunnel through that thick jelly coat that surrounds these eggs. It's pretty cool. There's even cooler stuff to come. Give me a minute to set it up, and we'll go to it. All right, so here what I've done is I've just taken that same sperm that I just left off with, and I've just moved it up on the screen, just so I have a little more space to draw some more. So again, here we've got the acrosomal vesicle has fused with the cell membrane of the sperm, and it has dumped out into the periphery here those digestive enzymes. And these actin filaments are polymerizing in this direction, and it's going to extend this finger-like projection, and that is called the acrosome. So here it is. So it's extended it further. So these are the actin filaments. Again, actin is just one of the cytoskeletal elements found in cells. Here's this is the digestive enzymes. And again, those enzymes are going to help it chew through this thick jelly coat that surrounds the egg. Now, one of the cool things is that and one of the things I haven't shown you is coming up next. You ready? Okie dokie. Get rid of that. Okay, so this one is the same as this. I just moved it up. Okay. So here's the same one, moving towards the egg. Check this out. Ooh, we see a new feature. So it's made a little progress through that jelly coat, and all of a sudden I'm showing you these little red circles here. I didn't show them before for clarity, but give me a second. Let me just close these out for a second. I'll go back to the original sperm anatomy slide. So when I was doing this with you at the beginning of the lecture, I didn't show you that inside of this acrosomal vesicle are these membrane proteins. Remember what an integral membrane protein is? I'm going to draw them in white just so you can see them. In the other drawing, I have them in red, but they're the same thing. So they're these membrane proteins, integral membrane proteins, that face the inside of this acrosomal vesicle. And again, there's these digestive enzymes. The digestive enzymes do not cut these, they're not specific to cut off these membrane proteins that extend out into the periphery. So what will happen is if we've got this, once we get that, so technically if I look here, I'll draw them in red now, they would now be on here. Because this whole acrosomal vesicle, when it fuses to the cell membrane of the sperm, it basically takes whatever was on the inside and dumps it on the outside. Okay, and that's what you see here. So these are the integral membrane proteins, so they're tethered to the membrane. And guess what? No, really, guess what? There's a receptor. I know. There's a receptor on the vitelline membrane that's going to help them dock. Sort of like bringing in the cargo. And these proteins are called, this is sea urchin still, they're called bindin. And they are on that acrosomal process. This whole little thing here, this like finger-like projection, is called the acrosomal process. Acrosomal process. And I'll do the drawing for the next stage in just a second. But again, these bindin were in that vesicle the whole time. It's just when the acrosomal vesicle fused, okay, they became exposed on the exterior part of this acrosomal process of the sperm. And we're going to get some recognition happening right here. Let's take a look at what that is. Isn't this so cool?